Today we will discuss about some of the cell mechanosensor that can triggers change in the biochemical pathways within change in the biochemicals pathways or signals within the cells because of the mechanical response that they experienced either from the extracellular matrix or uh, from the intercellular parts of the cells. So here they have shown one example, that is cell matrix adhesions, and we can see that when the cells interact with this fibrils, it leads to the conformational change, and that conformational change leads to interactions of the intercellular proteins such as vincilin and taline, and at the same time the extracellular part of these integrins will also interact with the fibrils, and this will cause the stretching of the integrins, or we can say the conformational change of the integrins, and that will generate some traction force. And in return, what will happens within the cells, here we can see that in the zoom in VH there is a actin elongations occurs, or which is known as ectomycin contractions, and these ectomycin contractions will then help towards the generating the traction force that helps in the migrations of the cells. And apart from these interactions, that is integrins interactions, there is also another interactions occurs between the cells interactions, and that interactions are basically because of this catherine on the one cells interact with the catherine on the another cells, and this also cause ectomycin contractions that can be seen here, that leads to the conformational changes. And these ectomycin contractions occurs in both the cells which are taking part in these cell adhesions, and simultaneously this nucleoskeleton pulling force will also observe in one of the cells which are moving in the directions towards the other cells. So here this cells on the right hand side is moving towards the left hand side directions, so we can see that the nucleoskeleton pulling force are observed in this cells towards the left hand side. So these are two major cell mechanosensors, and there is one more interaction, so one more example is shown here, where we can see that the integrins are interact with this BCR, and similarly not just one cells but other cells are also interact with the BCR. So this is the integrins interactions with the extracellular matrix, and that will generate this cell adhesions pulling force, or we can say that these integrins will cause the cells to move towards left hand side directions, and these cells will also helps the neighboring cells to move in that directions because of the cell adhesions pulling force, which is just now described in this panel A, how this pulling force move in the directions of their former cells. And one more thing, and so these are two examples that are shown here. So what happens in the pancreatic cancer, in any cancer cells, is that there is a extracellular matrix or tumor microenvironment are being generated by the cancer cells by secreting some of the cytokines or other interactions that cause generations of more collagens around the tumor cells, or collagen rich environment around the tumor cells, and that will uh, helps the cancer cells in not only the invasions from the immune system but also developing the mechanical changes, also developing the biochemical changes because of the mechanical interactions with the tumor microenvironment, that will have more of these mesochemical type characteristics, or that helps them uh, that that helps them towards the metastasis or migrations to different part of the patients. And this is just a basic overview about the impact of on the mechanical signals on the biochemical changes. If you need to understand more about this topic, that is mechanosensors and cell mechanosensors in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.